In this podcast, we're going to talk about what mutations are. We're going to talk about the different types of mutations. Then we're also going to talk about the effect of mutations at the cellular level as well as at the full organism level. All right, let's start with our definition of what a mutation is. So mutation is a change in the DNA nucleotide sequence. So remember, nucleotides have a phosphate, a sugar, and a nitrogenous base. It's actually that base. So if we put in the wrong nucleotide with the wrong base, that's going to change the nucleotide sequence of the DNA. So if that happens, it may alter the sequence of amino acids. And if you have a long chain of amino acids that are going to fold into a protein, that may also alter the structure and therefore the function of a protein. So it may not work correctly. And we discussed this a little bit back in proteins. So your DNA is pretty amazing when it replicates itself. There are proofreading mechanisms in place. In fact, only one error for every 10 billion bases usually gets through. And I'm going to age myself telling you this, but this makes me think of those typing tests that we used to take where you had to type a certain number of characters correctly, accurately in a timed period. And so however many you could type was your typing speed, your words per minute. And so I think about if I were to type 10 billion bases quickly, would I be able to get away with just having one error? And the answer is, oh, heck no. I am not that great at typing, but that's how amazing your DNA is. Not many errors sneak through those proofreading mechanisms. It's pretty accurate. There are chemicals that can induce mutations. We call those mutagens. X-rays and UV light are also considered mutagens. So if you expose your DNA to certain chemicals or X-rays or UV light, read tanning beds, you are going to increase the number of those errors that are going to sneak through your DNA. If we have a mutation in a somatic cell, the altered DNA is limited to that particular body cell. The only way you're going to pass that on is if that cell with that mutation goes through mitosis. Say we reproduce that cell, it's a body cell, a skin cell, and you need to make a new skin cell. That mutation will get passed on to the daughter cell, but it's kind of localized. That mutation is not going to be in every single cell throughout the entire body. It's localized to just that nucleus and any daughter cells that are produced through mitosis. However, if you have a mutation in, say, your machinery for going through mitosis, all cells have kind of an on-off switch. And if you have a mutation that affects that, where this cell reproduces and it doesn't shut off and wait, and it just keeps reproducing and reproducing and reproducing, what you're going to wind up with is this large section, this mass of cells with the same mutation, and it's going to start to grow uncontrollably, and all of these cells are affected by that mutation, and that's what we call a tumor, which can lead to different stages of cancer, so it can be very dangerous. If we have mutation in a sex cell, say our egg or sperm are carrying a genetic mutation, your altered DNA is going to affect the new individual because your egg and sperm are going to come together to create an individual, and then every single cell and every single nucleus that's produced from that original zygote is going to have that mutation. When that happens and every single cell of the individual is affected by this mutation, we call it a genetic disease. All right, let's go through some types of mutations. The first type of mutation is a point mutation. This is where one base in your coding sequence of DNA is substituted with another. So we're going to use these sentences to try to help you understand the different types of mutations. So this first sentence represents our normal gene. As the man saw the dog hit the can and looks like itis. Okay, so it doesn't necessarily make sense, but this was your normal gene. In a point mutation, we are going to substitute one of those nucleotide bases with another. So here in our point mutation example, we swapped out the letter G with the letter T. So in this case, it says, as the man saw the dot hit the can end. Okay, so we're substituting one for the other. In this situation of this sentence, you can see that it changes the meaning of the sentence. And a point mutation can change what protein product is made from that as well. A frame shift is where a base is added or deleted from the normal order. These are a little harder to pick up on. Okay, so in a frame shift in the bottom, and again, we're referencing the very top sentence as our original, we see that there was a letter omitted right where this bursting red burst is. And so the letter D was omitted, it was deleted. So as the man saw the og, it, heck, ain, notice that we're reading in threes, those are codons. So this shifts the reading frame over, and now none of them make any sense. These are the most dangerous types of mutations that you can have. So let's put this into words. A deletion is one or more nucleotides being lost from the DNA molecule. So they're being completely removed. So here we have, this example here was a good example of a deletion. The example that they're showing in the image, we have an entire codon being removed from that sentence. And again, that is going to change the meaning of the sentence, and therefore it's going to, if you were to connect it back to amino acids, if you removed an amino acid, that's going to change how your protein folds and will ultimately affect the structure. An insertion is where you have one or more nucleotides being added to your DNA. So here we have an entire codon being added to our original sentence. So as the man saw the fat dog hit the can. So we've added a complete codon here, or a complete word, which is analogous to a complete codon. So we're adding in a whole amino acid. Again, it's going to change the primary structure of your protein, and that's what you really need to get from this, is that when we add in nucleotides or we take them out or we substitute one from the other, we're changing the primary sequence of the protein, which is going to have an effect on the secondary, tertiary, and quaternary structure of the protein, which is going to affect the function of the protein. All right, let's talk about the effects of mutations or changes to your nucleotide sequence. There are three that I'm going to hold you guys responsible for. The first is a silent. So we're changing our codon, but it still codes for the same amino acid. So it's as if nothing has happened. The primary structure of the protein is intact. So here we have the DNA, no mutation. Our bases are TTC. In a silent, and that will code eventually, if we were to transcribe that to mRNA and then build our protein, that would be lysine. In a silent mutation, we have swapped the last C for a T. This is a silent mutation because TTT also codes for lysine. So we haven't changed the amino acid sequence. In a nonsense mutation, the codon that gets changed to a stop codon. So suddenly, you're reading along on your protein and prematurely you get to a stop codon and the protein stops being created at the ribosome. So here we've changed the first T to an A, and that codes for a stop codon. So no matter where this is, translation is going to stop and your protein will be incomplete. In a missense mutation, your codon is changed to another amino acid. So our original codon was TTC. Here in our missense mutation, we changed the second T to a C. This now codes for arginine. Or we could change it to a G, and it codes for threonine. Either way, you're sticking in another amino acid, which is going to affect the secondary, tertiary, and quaternary folding and structure of your protein. All right, let's talk about genetic diseases that are a result of mutations. So in a genetic disease, as we kind of discussed earlier, you have a mutation that's going to cause some sort of protein deficiency, or it's going to cause needed protein to be defective. An example that we keep bringing up in class is sickle cell anemia. Sickle cell anemia is the result of a point mutation. So we have one nucleotide base substituted with another. What this does is it alters the amino acid that that codon codes for, and therefore changes the primary and secondary and tertiary and quaternary structure of red blood cells. So instead of being nice and round, they have a half moon or a sickle shape. And this leads to all kinds of health issues because this particular shape of red blood cells not going to flow very freely through your round arteries. They get stuck. You're going to get blood clots. It's painful when you exercise in there. You don't carry oxygen as well as a round or not defective hemoglobin. Cystic fibrosis is another example of a mutation. So it's one of the most common genetic diseases in Caucasians, and it is a mutation resulting in a defective protein. So your cystic fibrosis transmembrane conductance regulator gene becomes defective. You need this gene for the proper passage of ions across the cell membrane. If you have cystic fibrosis, you're going to have decreased secretions of pancreatic enzymes, and this ultimately is going to result in a failure to thrive. So you're going to have very poor growth. Individuals with cystic fibrosis are also going to produce a really thick and sticky mucus in their lungs, so they're going to get higher levels of lung infections in this population, and also a shortened lifespan. So my little PSA for you, a lot of us carry a recessive cystic fibrosis gene and do not know it, so I strongly recommend you go get a blood test just to know if you're a carrier, especially if you're looking at having kids. Find out if you and your significant other are carriers to see what your chances are of passing that along to your children.